Hello, everyone. Good morning or afternoon or night, wherever you may be in this world. I'm Joshua Stein, Project Manager at the International Wheat Genome Sequencing Consortium, and it is my pleasure to serve as moderator for today's webinar series. So welcome, everyone. And today we are joined by Taner Sen, who will be speaking about the USDA GrainGenes database, which serves the small grains communities, including wheat, most of you may know. So before we go on to Tanner's presentation, I'd like to give a quick overview of the IWGSC. We are an international nonprofit consortium with 3,450 general members coming from 943 institutions and companies and located in 76 countries. And at this time, I'd like to take this time to acknowledge our sponsors who make this webinar series and other activities of IWGSC possible. So thank you to Arbor Biosciences, BASF, CIMMYT, Florimont Desprez, INRAE, Kansas Wheat, RAGT, and Syngenta. So thanks to our sponsors. As most of you know, the IWGSC was instrumental in the production and release of the first bread wheat reference sequence in 2018. Our mission since that time is to develop additional tools and resources to enhance breeding through an increased understanding of the molecular basis of traits and their allelic diversity. And to this end, I'm very happy to announce initiation of the IWGSC Wheat Diversity Project, to sequence 12 landrace genomes that encompass the worldwide diversity of bread wheat, update the Chinese spring reference sequence, and complete other aims listed here. And this is being done with funding from the National Science Foundation, the European Research Council, and is an international collaboration of scientists at INRAE, University of Paris-Saclay, and University of Georgia. So stay tuned for more information about this project as we proceed. I am also happy to announce as many of you know, the IWGSC holds a workshop every year in San Diego at PAG. This year, we are very fortunate to be able to do a workshop at PAG Australia. So that's taking place in September this year. And I'm very happy to announce that our call for abstracts is open. And so I encourage people who are interested in speaking at this workshop and traveling to Perth to please submit an abstract. The deadline is the 31st of May, and we have a link here to go to upload an abstract. And I also want to mention that we are offering a early career award to those that are still postdocs or PhD students or are otherwise defined as early career. And this will include a travel stipend. So I encourage people to go, and I will see you there. So just to give a quick overview of the webinar dashboard, um, uh, you can ask questions throughout the presentation, but we will be responding to the questions at the end. Depending on how many questions we get, your questions may not be asked live, but Tanner has kindly agreed to respond to all questions in writing, and we will have a link to those as well as the recorded video posted at both the IWGSC YouTube channel and at our website, wheatgenome.org. If you experience a technical issue, please use the chat. Also, please monitor the chat to see messages from the organizers. And you should be able to, at least by the end of this webinar, you should be able to download introductions and PDF formats of Tanner's presentation. So that with that, let's all please welcome Taner Sen from the USDA-ARS. He's also adjunct professor at UC Berkeley, And today he'll be talking to us about GrainGenes, a centralized nexus for small grains, data, and communities. So thank you, Tanner, for sharing your work today, and thank you all for attending.
Well, thank you, Josh, and thank you, IWGSC, for having me today. Today I will talk about many short topics, but to start my presentation, I want to take a step back and talk just a little bit about our challenges in sustainable agriculture. So one of the big challenges we have is the population increase. Right now, the world population is at 8 billion, and it's increasing at a rate of 1.1%. So that creates many challenges, including food-related challenges. How can we feed this population? And the second part is the food security as far as the yield goes. We want to increase the yield so that we have enough to feed people. But we have insufficient yields right now, so we need to increase the yields. On top of that, the arable lands are decreasing. So that's two constraints and at the same time. But just increasing the yield doesn't ensure the nutritional value. So enough food doesn't mean nutritious food. So we need to also breed according to targets that can have a nutritious food. So this in itself is like a bunch of challenges that are like hard to surmount and we need to work towards. But on top of that, we do have a climate change affecting everything. And it affects different areas differently. Sometimes we'll have some areas that will have higher yields because of climate change, and other areas we have lower yields because of the change. So we need to think these challenges within the context of climate change. And I want to make 2 points. One is that the burden of this is not shared equally across nations and groups. So we need to be mindful of inequalities that is creating. So maybe in certain areas, we can have more yield, in other areas we don't. So I think it's important for us to think about what kind of inequalities there are. And also, and maybe it's a good thing that this is not just one country's burden, but a global one. So it requires global policy solutions. Of course, we're scientists, most of us. I don't know who joined this presentation, but the policies, of course, we don't have much say, but we do have say about the research. So I think through collaborative research across nations, we can solve these issues. So this gives us an opening to respond to these challenges. So definitely, we need then do more research to address agricultural challenges. And when I say we have plant geneticists, plant breeders, data scientists, soil scientists, programmers, entomologists, and agronomists, curators, and competition biologists who will need to work together to address those challenges. One thing that actually these the different experts are communicating through, and a medium that actually that can be useful for our research is that they're all generating data. So data between these expertise, between these studies, is the connective tissue connecting these different studies so that to address agricultural challenges. And so we generate data, our groups generate data, the researchers generate data. So how can we increase the value of research data even more? So let's say that we have 3 data sets here. The first one is that publications, and there's maybe some data in the supplementary material. And data set 2 is the data deposit at Figshare. Okay, and these are good things that we are actually making data available in some shape or form. Data set 3 is actually in the student's computer, and may or may not be available going forward. although this is good, we're generating data, we have to think about the gaps between these data sets. So if it's not linked, not connected, is it accessible? Is it findable? Or is it just lost? It's just data is somewhere, but we don't know what it is, and we cannot access it. So I think it is clear that the linking and associating data is important. I want to explain this just with one example. So the value of linked data. And this example I will call Japanese. Japanese is a restaurant in the San Francisco Bay Area. It has been around over 50 years and one of the contributors to the California cuisine, actually. And it is also part of the popular culture. If you're aware, there is this director, Werner Herzog, and he made a bet, he lost it, and he ended up eating his shoe. Not the sole, it turns out, but the shoe itself. So Japanese restaurant is very important. But you can think about a restaurant that is near you that is highly regarded. Okay. So I'm hearing about Japanese. So what can I learn about this? Right? So I can go to different data sets to learn more about this restaurant. And maybe there's a data set that says restaurants, and there's Japanese right there. And maybe there's a menu table that I can go and then learn more about the restaurant. Maybe I can look at the reviews, right? And then I need an address to go there. So as you can see here, there is no linkage. There are just data sets, databases that are separate from each other. But we know that when you link these data sets, actually it becomes very useful. So if you go to Google, you put Japanese, and then you have the all kinds of information that is in front of you. And in a sense, that's what you want from your biological database as well. Of course, we're not Google, but you want that connectivity, you want this linkage, you want this findability. So that actually tells you about the value of a sometimes centralized location that provides linked data sets and tools to users through the same website or browser. But that doesn't necessarily be a centralized location. There can be different locations, and then you can connect those. The value is in the connections, essentially. But centralized locations really help because it tells you where to go. We go to Google to do searches. In that sense, centralized locations for biological databases is valuable in itself as well. So GrainGenes is one of those centralized locations. So we are around for the last 30 years. And this is our URL link. And if you go to this URL, you will see our homepage here like this. And our users are globally located. And we serve small grain researchers worldwide. I mean, this just shows that we have users from Americas, Europe, India, China, Africa, Australia, so all over the place. So we are here to serve the small grains researchers, which includes wheat researchers, barley researchers, rye researchers, and oat researchers. But we like wheat a lot. So when you look at GrainGenes, we do have data, which I will talk about, and tools. We do research, and we work with partners. But in the middle is actually global small grains community. So that's our core. And we do have tools and services just help the community to become a community or to act as a community. What do I mean by that? One is, so we have community services, and the first one that comes to my mind right away, and most of the graduate students and postdocs, I assume, is the jobs. What's my next position? So if you go to our homepage, we have actually areas that you can go to job listings or jobs. And then if you click on it, you will be able to see some available jobs. And we do this as a community service. So advertisement cost is zero. So, you can just go to our website and check it. Or you can join our GrainGenes email lists. We have Grains email list as well as Oatmeal spread to run email list. So if you join this list, you will receive this job listings, sometimes community information, or community announcements, and so on and so forth. I put these listings on Twitter as well, and you can actually follow us from there. We also have tutorials. If you are using the database for the first time, or if there's a new tool that you're not very familiar with, you can follow the links under community services or under quick links to go to the YouTube videos. These videos, some of them are long, some of them are very short, but it tells you how to do certain things easily. And if you have any suggestions for us, let us know and we can create new tutorials going forward to answer user needs. We also have species portal. And this is an interesting area because we do have like annual wheat newsletter, barley genetics newsletter here, but also we do have like project pages that we provided to research groups, like Global Durum Genomic Resources here, as well as PANOT, which is the PANOT project, is the international consortium. We also have areas for specifically dedicated nomenclature, because we believe the importance of nomenclature, the importance of standardized vocabularies for our research. So we have an oat nomenclature area and Wheat gene catalog area. And Wheat gene catalog area is essentially the PDFs that were generated over the years by and then published that are the genes that are curated essentially. So we provide these services, and we are going forward. We work with the research groups to help them to create these areas so that they can share information about their projects. They can share some documents about the projects, and when they publish a paper, we put that publication there as well. In the middle, we do have hot topics here, right there. And as you can see, we put recently the Turgidum durum wheat Svevo version 2 pseudomolecules. So if you follow that, you can see what is available, what kind of data set is there for you to reach them. So let me talk about a little bit of a data content and tools. Well, before talking about data content, perhaps I should mention the FAIR principles, because this is what we are striving for in GrainGenes. And FAIR principles, for those who are not familiar with it, essentially FAIR stands for findable, accessible, interoperable, and reusable. And these are true for in terms of data, in terms of metadata, or the programs, scripts that you are using for your research. You can actually go to the FAIR data paper to learn more about this. And just thinking about how we can apply this principle so that data access is facilitated and the data discovery is facilitated is important. And if you look at the project planning, data generation, when you put data in a database and data publication, these are all part of the data cycle, data life cycle that FAIR principles can be applied to. And if we actually make data fair, we will increase the utility of data. And just think of Shepani's example that I gave earlier. Linking is easier with fair data. If linking is easier, then it's easier to find the data. And here also, of course, the acuity issues need to be taken into account. When somebody, for example, publish a paper, let's just start there. It will be best if that paper is actually open access. In the North America, for example, we may have access to papers through our universities, through our organizations, but this may not be true in every part of the world. So just making that papers openly accessible will actually open that research to the wider range of people, and that's what we want, right? And thank you, Ekbay for providing this slide. So I'm talking about data discovery. So let's see how in GrainGenes we can search for data and access data. So under quick links, we have search and browse GrainGenes. So if you click on this, you will go to this area. So on top, we have the GrainGenes class browser. We have a bunch of classes. Actually, the classes are here down here: allele, assembly, author, colleague, collections, so on and so forth. So you can actually do a query here and go, and then you'll get the results. If you want to go click on a specific class, you can do that too. You will have a list. You'll get a list of things, and then you can choose. You can go from there to see what kind of naming system we used or what is available. We put something new recently. We allow the users to search genome browsers. Now that we have over 100 genome browsers, that sometimes it's so difficult to just go one by one and to see and to search for a gene model. So this search capability, you can just put the gene or transcript, and it will tell you which browser is actually that gene is located. So that I think that this is useful area that users can actually benefit from. We do have a search GrainGenes with Google area. So these are for static pages. So you can put some search term and you can use actually Google itself to search our website. And last but not least, we have a search with IS area. So with IS is a WIT information system. It's actually under the created under the WIT initiative, and it's a collaboration that different databases share their data sets. So with this area, the keyword here, you can put something, a keyword, let's say just a protein, right? And then you will go to the WIT IS website, and then you can see the results from different databases. So let's say that the first thing is actually GrainGenes, and you click on it, and then you go to the GrainGenes website. And it's not just GrainGenes. There are different data providers like IPK, Grameen, and INRAE, and other places, so that you can go to their website. You can find those data there. So this is another way of linking the data. You can link the data at the database level. Also, you can link the data at that's API level that will allow the interface level linking possible. Of course, we have data, but data veracity is important too. That you want your data to be accurate. And in order to do that, you need curation. Curators are dedicated professionals, and they actually do so much for our communities. And I think that they are not getting enough shout out for that. So thank you. In our GrainGenes, we have Victoria Black. So we are very grateful that she has been doing curation for GrainGenes for decades now. And here I'm showing the curation for some of the genetic data sets. And this is one of the strengths of GrainGenes. Although today I will mostly talk about genome browsers, genomic data, and I will talk about BLAST, which is a sequence alignment. One of the strengths of GrainGenes is the genetic data, like genetic markers, genetic maps that we have. And we continue doing this, the curation in these areas. For example, here we did a StripeRust QTL curation, or different gene curation into GrainGenes. We also created the GWAS results, for example, for pathology, salinity, and agronomic traits. We try to link the genetic data sets with the genomic context. So we created actually, or we use the data generated by other groups to create tracks for our genome browsers for this QTL. Or we use CMAP to show the genetic relationships in a genomic map context. So we try to go linkage from different data types as well, like this. As I said, we have tons of genome browsers right now, more than 100 at this point. We definitely, of course, have IWGSC. We work with IWGSC for the version 1 and version 2, and we have genome browsers and tracks for that. We have genome browsers from the Wheat 10 Plus project, and we just released the SWEVE 2, which is available under the Toronto Agreement. We have over 30 genome browsers for the PanOat project, and we are building the Barley Pangenome browsers, which is 76 of them. These are separate genome browsers, essentially, right? We do also have more integrated and showing comparative relationship browsers. This is, for example, the Einkorn pangenome area, and you can have access to different tools, JBrowse, BLAST, Weaver, and Synteny, showing Synteny areas. And so you can see a JBrowse tooling result, or you can have BLAST results, or you can visualize your BLAST results in a different way, and then you can use AcuSyn visualizations, for example. So we do provide pangenome-centric views, either like by single genome browser, browsers, through single genome browsers, or through comparative genome browsers. As I said, we do have GWAS QTL tracks in genome, and I just want to show one of them here. This is from a Barley example. So you can click on this, and then you can go to the QTL report specifically, and here you can see other types of information, such as gene class, marker information is available, and then you can see the trace it's affected, and you can click on this and go to the other sites to get more information. Or you can even click on this and then go back to the genome browser if you desire to do so. So there are different ways of entering and then using data in GrainGenes. One collection that we started working on is the Watkins collection. So I'll call this Watkins collection Plus. If you were at PAG this year and there was a workshop for Watkins collection, this is essentially a combination of land races and modern wheat cultivars. So it's over 1,000 accessions. And this is collected globally. This is a wonderful bioarchive paper preprint that, as I just said, you go and read. There's so much data and so much useful data. So from this data set, we started working on creating and integrating data into GrainGenes. And one thing we did is to create vaccine collections SNPs into the IWGSC reference, IWGSC's 1.0 genome assembly. So there was so much data that can be handled by JBrowse that we actually divided the SNPs into 2 tracks. So we have one track going from first to fourth, and the second one is 57. And there's so much information behind that. If you click one of the SNPs here, you will actually learn more about the SNP, that if there's a nearby gene that is controlling, and so on and so forth. So this is just some of the data that you can get by just clicking on that specific SNP. You can also go down and you can see other types of information that were provided by that's provided by the Watkins collection paper. In another tool, content/tool, we do have blast areas. This I'm just showing the wheat part. We have more, obviously, but this is for our nucleotides. And if you see a little symbol next to a database, that means that it has direct connections to the genome browser. Let me first go to the protein side. So you can choose either nucleotide and protein. And these are the proteins that were provided by the genome assembly and annotation groups. And you can actually run your data here. And again, if there is this GG icon next to it, it means that it is connected directly to genome browser. So linkages, let me talk about it. What I meant by this, well, hey, if there's an icon, what does it mean? So you, for example, put a sequence here, and so you run your blast, and then you obtain a result. So if you see here that there's a JBrowse link, that means that I can click on this, and I will go to a JBrowse instance where the hit is. And here, actually, hit is shown by that yellow area. Okay. And this is essentially helps to the FAIR principles to be applied, because you're actually connecting the data, making it more findable. Okay, so you can also go from the genome browser to BLAST, and this is something we developed in GrainGenes. You can rubber band an area, and then when you say click click BLAST, it will take you to BLAST with that area sequence already pasted in the BLAST, and then you can choose a database, and you can run your BLAST to obtain results. You can also use Genome Browser as a way to go to other resources. For example, in this case, we have this marker, and then if you click on this marker, you get, of course, description about this marker, but also there's a T3 link there. So if you click on this T3 link, then you will be going to the T3 toolbox database. So you can use this area to reach other places, like I did earlier with information system. But this is not the only link available through the Genome browser. We put links for XWeb, Netminer, Ensembl, and even Persephone. This is another area. This is something new. So for some genome browsers, we do have data that's associated with UniProt IDs, and we use this UniProt IDs to find whether AlphaFold predicted AlphaFold structures exist. So we created a track that says proteins with AlphaFold links. And if you click on it, then you can see that there's a clickable link here. And if you click it, it will go to the AlphaFold website. We did this specifically because not every gene model will have an AlphaFold link. So we just wanted to make sure to highlight the gene models whose transcripts or translated proteins will have a three-dimensional protein structure. Last but not least, I mentioned this earlier, nomenclatures and data standards are very important to facilitate data discovery. So there are two main publications in this area. One of them is the oat, recently published, but the other one is wheat. And so lots of effort went into this, and please use the guidelines that are provided here. And remember the Japanese example, or your favorite restaurants example. You want more data. When you search for data, you want more data there. And if we are using the same language, if we are using the same terms, then we will be able to find things easier. In this area, more work is ongoing on protein naming, and Cristobal Uau taking the lead on this. And genotype abbreviations will be in GrainGenes. What does that mean? For example, you're working on a new accession and you don't know how to name gene models. So these publications give good flexibility about choosing one, but you have to come up with how would you abbreviate the accession. So you can talk to me, you can talk to other people like Cristobal is taking the lead, as I said, and we will have a list in GrainGenes that you can check against to see if your accession, your genotype is already there and the abbreviation. If not, we can always add it so that the next person can use the same abbreviation as well. So this is data content, and let me talk about the research a bit. So this is just I think one slide that I put there, so I will just touch just very quickly. So we work on the protein phosphorylation area, and protein phosphorylation is of course very important in plant development and immunity and stress response. So we developed a workflow that involves large language models, and these to capture the modeling of proteins so that we can apply this to develop a better prediction methods for predicting phosphorylation sites. So as compared to other methods, our method called PhosBoost performs better than others and is comparable with the top of the phosphorylation prediction software while providing higher recall. So higher recall comes at the trade of reduced precision. So there's more false positives, but you get more true positives. So that actually increases your set, or if you want to go to the next step in the research of looking which phosphorylation site may be of interest. We predict more functional phosphosites and for the functional analysis. So another area that we worked on is the pan-intractome networks in maize. This is essentially the NAM populations in maize. There were 26 individual maize genomes, and we created interactomes based on the predictions for protein-protein interactions. And then we clustered those, and then we did annotations. So our aim was to create these clusters and essentially guilt by association. If there's a gene there that is in the middle of other genes that are part of the cluster and they share some functional similarity, perhaps the gene that is part of this cluster, which is not yet functionally identified as such, may have similar functions. So we published this paper. We have all the clusters available for different traits. So that's one thing about this population. We do have genomes. We do have all the proteins, and we also have trait information. So we were able to actually connect these two to be able to be able to look at the traits and trait specific clusters to deduce the predict the functional proteins. So today I present this, but of course I'm just the data was generated and deployed by a great team. It's a great GrainGenes team. Myself, Gerard Lazo, Victoria Blake, Steve Michael, Eric Yao, Eli Poretsky, and Parva Sharma are working together to create this data content and tools for the small grains community. So we have partners. These are just some of the partners. Definitely IWGSC. We work with AFC in Canada. We work with different consortiums like PANOT, Tetraploid Wheat Consortium, PepsiCo and Corteva in one of the companies. We work with other ARS databases, EggBiodata, and we're happy network. Definitely Wheat Gene catalog with Bob McIntosh, and Ensembl. We contribute to the International Wheat Congress. Definitely, we are very close with URGI, and we work with them at VTIS and to share IWGSC data sets. And of course, VTIS is connected to the FAIR system in Europe. So take-home messages. One is that we have extraordinary global challenges when it comes to agriculture, and it's shaped by climate change. So we need to put more efforts into research, but we need to then look at the data that we generate and make sure that they are fair, they are deposited at the right places and linked. And we are here to serve you, to provide information, to provide guidance, and to provide data content that is linked with other data sets. We do what we do because we work with this species because they provide food, fodder, and beverage, which means calories and nutrition. It means calories of nutrition for little kids, for agriculture, and also the big kids, which we are part of actually. So that's actually the last line. So please cite us. This is very important. It shows that at least people are using our website and publishing it. And we appreciate data generators, and especially the ones who work with us, and that's how we can make this available. And GrainGenes Liaison Committee, which is an advisory body to us, that actually provides us guidance. Please contact us for your needs. If you have questions, there's a feedback button on our every page actually. Just click on it and let us know. So thank you.
Thank you so much. I'm sure there's applause going on
Yay!
around the world.
It's over.
That was terrific. Thank you, everybody. So we do have a few questions. I'm going to start with some specific questions. This question came from Ahmed Sharif. He says, is there a database of mapped QTLs associated with different traits in wheat? And if no, are you planning to have such a thing in the future?
We have QTL data that is mapped to wheat. We have, so I don't know specifically for wheat, but for all, we have like 9,000 QTLs. There is also other QTL databases that the user can go and access. I think that's called QTL DB, but I'm not sure. If you just click on it or let me know if you have questions, I can give you that information.
I'm aware that you have QTL information and that you have curators that are going into the literature and trying to relate those map positions to the reference. And actually, maybe I'll bring this up later, but with a new reference coming out that might replace the existing reference in the next 2 years, it'll be very important to bring the community up to speed and do everything we can to enable that transition. So that's something that I hope to be working with you in the future,
Absolutely.
Tanner, and also getting input from the community as well on this to make a really good transition. Okay, here's another question. So this is from Mohammed Amir Khan. Thanks for the informative session. Is there any unique feature of GrainGenes that other websites don't have?
So I think we have definitely known for our genetic information, genetic markers and genetic maps. That's new. And we do have lots of genome browsers and BLAST databases that you can go to in a centralized location. And as I said, the value comes from the linking the data. So as a user, you go in, you do some research, like some search, and where you can go is important. Will be just stuck there, or if your data will be connected to something else. So as you said, we have curators actually that pay attention to these things to make sure that it's not just uploading data, but also relating data to the existing data. As I said, GrainGenes is around since 1992, so 30 years. Lots of genetic data is available.
Okay. And here's a question from Pat Heslop Harrison. Has anybody created a student practical class based around the amazing GrainGenes resources?
Yes, actually, Victoria Blake created a student module for a class, which is very curation centered. So that's one thing, and we provide sometimes like a tutorials, but we've realized that instead of giving a tutorial just like 10, 20 people, uh, we create these YouTube tutorials so that more people can access to it anytime. So that actually helps the like people who cannot come to our tutorials to learn more about what they can do with GrainGenes. So I suggest that you go to the tutorial videos and that may be actually part of the class modules, and let us know if you use in your class the GrainGenes modules or tutorials.
Okay, there's additional details to the question that maybe you could delve into further offline
Uh-huh.
if you'd like
Sure.
um, on this. Okay, here's a question. Okay, from Andrew Reed, the Shea Pani.
Ah, yes,
The yeast
Japanese.
example is really helpful. Can you use this metaphor to help me understand how T3 and GrainGenes interact?
Well, okay. Maybe not with Japanese, but so T3 works with we are complementary, okay, and we have weekly meetings, so we work very closely with them. Uh, they are focusing on the field trials, genotype and phenotype data while the trials are ongoing. When the data is published, then we actually look at the peer review. So it becomes a peer review publication. And then we do not deal with sometimes the raw data, but process data, what is obtained. And then we put that data set, those outcomes into GrainGenes. And the genome browsers actually, I should have mentioned this. We of course definitely they're located in GrainGenes, but that's actually a concept T3 genome browsers. They create tracks on these genome browsers and then to give links to other T3 pages. So we work extremely closely and we made the decision not to recreate the resources with genome browsers. So we use shared genome browsers with T3. So Japanese, I mean, maybe the food is coming on the way, and then when the food comes, then that's the GrainGenes, I guess. If they're cooking it and then bringing it table.
Okay, here's another question from Marina Martinez Garcia. Many thanks, Tanner, and group for all your work and efforts to bring accessible data to all. Do you know if there's an updated way to connect map disto QTL intervals directly with GrainGenes browsers?
Um, That's I don't know off the top of my head. Is this something that CMAP can handle? It is possible. I'm not sure with that technical term. But we can look into it and then I can get back to you to her, I believe.
Okay, great.
Later on. Yes.
Okay, that sounds great. And then we have a big thank you from Herman Buristemeyer. Um, so that's a very nice compliment. Great. Okay. Okay, here's a question. Thank you very much for your nice and informative presentation. Is there any room for doing parts of research in your laboratory? Can you support sequencing of barley genotypes?
Uh, yes, we do the research. Actually, I we have a postdoc position open, which on the like more like using computational biology, of course. It is available. We work with the barley groups as well. We do have a barley project. The we do not do sequencing. sometimes usually consortiums nowadays we actually have a sequencer group that provides that service. But yes, we I can lead that person to the job ad, and then we can keep the person in mind when there are new positions open up. Just send it feedback. Use the feedback button to send me something, and let me know, and I can direct that person to the job ad.
Okay, we have about 9 minutes left. There's questions in the webinar chat that I'll try to get to. This is from Roberto Tuberosa. There,
Oh, Roberto.
there are many examples of QTL-ohm meta QTL analysis. It could be good if GrainGenes could somehow collect such meta QTL analysis. And thank you for a great job.
Yeah. Thank you. Yeah, thanks for bringing this up. Roberto definitely is the person who actually in the group created QTLs for Drum Wheat. So we are very appreciative that of that fact that they took the QTLs and aligned it to Drum Wheat. I think that's an excellent idea, Roberto. Let's talk offline, and definitely we can put some effort into this to put more metadata into QTLs and look at what kind of analysis we can do. And yeah, as you know, metadata is very, very important for to make data fair. Because without metadata, we won't know what the data is about, right? For those who are not familiar with this data science terms, metadata is data about data. So if you collect something, then you have all this information about how you collected it, what kind of collection data types you have, etc. So it's very, very important. So yeah, definitely. Let's let's connect and let's talk about this offline.
Okay, here's a question from Sean Yarns or Yarnez. Is GrainGenes BRAPI interoperable?
Not yet. We are working on it. We just like recruited someone to work on this summer. So she will be creating some interfaces for BRAPI compliance, for BRAPI discovery. We definitely work with Paul Selby for a different project, but we didn't make yet GrainGenes BRAPI compatible. But we think that this is the way to go. Definitely, BRAPI is very valuable resource. You can use BRAPI to reach other networks in America, in Europe, and other places. So yes, we will be working on this.
Okay, and Marina Diaz, could you recommend a tool for expression analysis in wheat? I know wheat EXP and EXP VIP. Very
Mhm.
interesting webinar. Yeah.
Yeah, XWIP definitely is important for the expression analysis. Uh, uh, in Toronto, I think Provar is creating expression views, which are beautiful views that he actually puts the expression on the organs, the different development stages. So Nick Provar is very good in that. And after that, if you want to go to look at the networks, then Netminer is the step to go after what you want to do. Like I have all these overexpressed genes. How do they connect? And then you go to Netminer. But good
I,
question.
I believe Ensembl might also have some expression atlas
Oh,
data for wheat, so the uh, might want to check that resource too.
okay. Yeah.
Okay. All right. Oh, wait, I might have missed one. Oh, okay. This is from Tanushri Halder. I'm wondering about the IWGSC RefSeq version 2 has complete protein information which may be useful for proteomics data of wheat.
uh, yes.
So you have the browser for version 2 and
Uh-huh.
the gene predictions and protein sequences available for that. Um,
Yes, as provided by the IWGSC.
yes.
So we did not create this. So let's be clear about that. But yes, we do. Once we if the genome assembly groups annotate and genome assembly annotation groups provide this kind of information, we just put it up as in the genome browser or on our blast. so that you can use this kind of information. And,
And yeah,
yeah.
and I believe also there's if you can't find information at GrainGenes, there's also the if you look online for the IWGSC repository with the release of version 2, there's some companion data that relates version 1 to version 2, things like gene ID mappings,
Oh, the
things like that.
correspondence table. Yes.
Yeah, might be very helpful as well to
Yes.
researchers.
Yes.
Okay,
Exactly.
we're running a little low on time. There was one other person I want to really thank, Jun Hu, for attending. He's a chemist. He had a number of very general questions that really didn't relate specifically to grain genes, and I've offered to start a discussion with him or her on those questions uh, uh, by email, and I'll also share them with you, Tanner.
Oh, thank you.
Okay. All right, well, we're running low on time. We have just less than 3 minutes. Um, okay, there's a couple of more questions that I didn't get to. I apologize, but I believe Tanner will be able to follow up with everybody
Absolutely.
on all of these questions. Again, I want to thank Tanner for a great, informative webinar, and I want to thank all of the attendees for joining us today. And it's a great community, and thank you so much for joining us today.
Thank
And
you.
with that, I'll say goodbye. Thank you again, Tanner.